Thank you and welcome everybody. It's a pleasure for me to be here, and I'm honored to give this webinar. This is going to be an overview of some recent studies that I was involved in, and we're trying to assess the metabolic profile of individuals with autism spectrum disorders and other neurological disorders. So we use this approach. I hope you can all see my screen now. We use this approach to try to answer some of the new challenges that are posed by the post-genomic era. So we are now facing the problem of dealing with a larger set of data that are more difficult to interpret. And since we have the technology to generate the data, now we need more functional validation of the genetic variants that are detected by these next generation sequencing. And we need to utilize and to explore new functional approaches to investigate the pathogenic mechanisms associated with these variants. Also, another challenge is the definition of the genetic component, the genetic predisposition for complex disorders, and particularly to assess the genotype-phenotype correlation in various conditions characterized by high variability. The important thing to consider is that one of the challenges of this post-genomic era is to deal with a much larger number of conditions that have genetic components, because we're no longer talking just about the rare genetic disorders, but we're talking about very common disorders that are complex by nature. So they are caused by both genetic and environmental components. And one of these disorders is, in fact, autism spectrum disorder, or ASD. You can see here the three main core symptoms that are necessary to make the diagnosis of ASD, and they involve deficits in social interactions, impairment in communication, both verbal and nonverbal, but also restricted repetitive patterns of behavior, activities, interest, and so on. This is a relatively young disorder as compared to other frequent conditions. Has been described about almost 80 years ago now, but the definition has been constantly object of reconsideration, and the latest one is the one by the DSM-5 in 2013. The incidence of autism has been growing dramatically in the past decade, and the most recent estimate is about 1 out of 54 US children at school age, so around 8 years of age. Also, the other important challenge is that 80% of cases remain idiopathic, which means they don't have a very well characterized cause. And the symptoms must be present in early developmental period. So this is a pediatric disorder, but there is a problem because the average age of diagnosis for individuals with ASD is around 3 years of age. And that is because the diagnosis is mainly based on behavioral observation, as you could see deduct from the three core symptoms. And there is no preclinical screening, no lab tests, so molecular tests that could either validate the behavioral diagnosis or help to diagnose the borderline cases. And sometimes a late diagnosis will be a problem because it will prevent the enrollment in early intervention programs. If we look at the etiology of ASD, one of the most striking features is the male to female skewed ratio. The most recent estimates are about 4.3 males per 1 female. But we know there is a high heritability based on twin studies, so the difference between the prevalence of ASD among monozygotic twins versus dizygotic twins, and the estimates of heritability in ASD is about 85 to 92%. There is also high recurrence in siblings, and as you can see, not only it increases with the number of siblings, but it is also influenced by the gender, with higher chances in case you have a male offspring. So all these epidemiological features point towards a strong genetic component of autism. And in fact, genetic abnormalities have been largely described in the cohorts of individuals affected with this disorder. There are about 1,000 genes that have been associated to different degrees with ASD, but only 1 or 2% of cases have causative single gene pathogenic variants, which means that individual sequence variants that are well classified as pathogenic, therefore causative of the disease, in just one gene have been described in a very, very small percentage of individuals with ASD. On the other hand, about 1/4 of the cases are estimated to carry some genetic variants that would work as modifiers. So will exert some effect in either the onset or the severity of the phenotype, the behavioral phenotype, but will not be sufficient alone to cause the disorder. So we use the biolog phenotype microarray mammalian plates to approach the functional study of individuals with ASD. This is a test that is based on the assessment of the energetic response in different metabolic environments. The energetic response is measured via NADH. There are 96 whole plates, as you can see here, and you can see the different color because there is a specific redox dye mix based on tetrazolium that reacts with the amount of NAD that is reduced to NADH. So the more NADH is produced in each well in the presence of different compounds, the darker the dye will turn in that well. And you can see here the parameters, some of the parameters for this assay that will include a 24-hour incubation with the dye that will allow for kinetic reads, as you can see here, where we have red as control, green as patient, and yellow is the overlap. So we can see not only the final production of NADH, but also the progression, the kinetic progression of the production in each well, and we can compare individual cases versus controls or entire cohorts of cases and controls. As I mentioned, there are different compounds in each well, and the plates from PMM1 to PMM4 include energy sources that could be carbohydrates, carboxylic acids, or amino acids, and plates PMM5 to PMM8 include metabolic effectors such as ions, hormones, cytokines, and other molecules. So the big difference is that in the first 4 plates, the cells utilize the compound as energy source. So there are plated with modified medium that includes fetal bovine serum. In the plates 5 to 8, they are plated with the medium that does not include the serum, but does include energy source, which is preferentially glucose. And we measure the amount of NADH the cells produce and the utilization of that glucose, but in the presence of different metabolic effectors. So we measure the capacity of the cells to be energetically effective in different metabolic environments. And by using this approach with the cohorts of individuals with autism, we were able to see a clear difference in the utilization of the amino acid tryptophan as an energy source. Here you see the average values for 20 individuals with autism that are in blue and 20 controls that are in red. And this is for plate PMM4 that includes several dipeptides. The wells from E7 to F6, where you see this clear change, contain dipeptide with tryptophan as a first amino acid. So we explored further the possible relationship between tryptophan and ASD. And first of all, tryptophan is one of the 8 essential amino acids. So it must be added, it must be introduced in our body through the diet. The absorption rate, therefore, of tryptophan, so the availability of tryptophan for our organism, depends among other factors, on the bacterial strains in our intestinal flora. So there is a link to the microbiome, which is the genome of our bacteria, and the microbiota, specifically the one in the intestine. And in several patients with ASD, abnormal strains of microbiota have been described in the intestine. Tryptophan is also the precursor of several neuroactive molecules, as we will see in the next slide. And there are studies that show that depletion, both acute and chronic depletion of tryptophan, has been associated with mood disorders. So as I said, tryptophan is the precursor of serotonin, which is one of the major mood regulators, but is also important in the early stages of brain development. But also through the serotonin pathway is the precursor of melatonin, which is the major regulator of the circadian rhythm or the sleep-awake cycle. And abnormal sleep patterns have been described in patients with autism. Following another pathway that actually utilizes over 99% of the intracellular tryptophan, this amino acid is associated with the IDO1 and 2, which is one of the most potent endogenous inhibitors of our immune system. And it's critical in early stages of tumor development, but also in the tolerance towards the embryo. And it's very important to extinguish the inflammatory response. So again, there is another connection between tryptophan and some of the features reported in ASD, where immune disorders are relatively common. And this pathway leads to kynurenine, and then the end products of kynurenate and quinolinate, which are antagonists to each other, and they're very important. Their balance is very important in their development, because kynurenate is neuroprotective, quinolinate is neurotoxic, and it's involved, as you can see, in mitochondrial function, and also it is important in early processes of neuronal apoptosis. And finally, quinolinate is a structural precursor of NAD, nicotinamide adenine dinucleotide, which again is NADH in the reduced form, and therefore is linked to mitochondria, as we said, dysfunction, through the availability of NADH as energy carrier for the respiratory chain. So as you can see, tryptophan is an amino acid that may play multiple roles and may be associated to ASD through several pathways. Therefore, we decided in collaboration with Biolog to develop a custom-made plate focused specifically on tryptophan. As you can see, we have basically one unit of 8 wells vertically that is repeated 12 times. In this unit, we have glucose as a positive control because it always provides a strong energy source. We have an empty well, so no energy source provided, which is a sort of a negative control. Then we have tryptophan alone, and then 5 dipeptides out of the 12 that we saw being so informative in plate 4, where tryptophan is the first amino acid. And of course, we started with the same conditions that we used in the Biolog protocol for plates 1 to 4, because these compounds were analyzed as energy sources. And as you can see here, we have some repeats. These are 2 individuals with autism. We used lymphoblastoid cell lines and 2 controls. And we repeated, we have 6 repeats for each sample. And you can see immediately, just by the color in the wells, that controls produce a stronger signal, which means a higher level of NADH in the presence of tryptophan. And you can see here in the kinetic graphic how there are not many striking differences in glucose, some differences in the empty, but a consistent difference in all wells containing tryptophan, particularly in the endpoint. So there was not a major difference in the slope. So the kinetics of the NADH production in the presence of tryptophan as energy source was not particularly different between patients and controls. But the plateau reached by the cells was different. So the cells from individuals with autism stopped earlier their production of NADH. And we published these, and here you can see the data generated using these tryptophan plates on 50 lymphoblastoid cell lines from individuals with ASD versus 50 controls. And you can see the significant differences in all wells containing tryptophan. But we noticed that if we wanted to use further this assay, there was some margin for improvement. Particularly, we wanted to increase the capacity of the assay to sort out ASD from control, so to increase the discrimination power of the test. We wanted to optimize the test parameters and to minimize the background noise, particularly related to the empty well. So we were still aware that there was a problem in non-specific NADH production. So by doing this, we explored possible alternatives for the parameters. First, we looked at the protocol we used to culture the lymphoblastoid cell lines, and we noticed that there were no significant differences in NADH level in the final outcome when we changed their feeding schedules. Then we looked at the FPS, so the fetal bovine serum type and concentration that we used, and we explored different concentrations, 1, 5, 10, and 20%. 5% was the original parameter in the protocol. But we also tested two types of serum, the dialyzed fetal bovine serum and the regular one. The dialyzed gave us no signal, so it was too weak for the system. The fetal bovine serum, we noticed that increased concentration of fetal bovine serum was associated with increase of the signal, but also increase of the background noise. Then another parameter we checked was the number of cells per well, so the concentration. The starting one is 20,000 live cells, and we explored the lower concentration like 5,000, 10,000, but also higher with 40,000. And we noticed that as we increase the concentration, we increase the signal, and we decrease the variation within the cohort that we analyze. And then we tested the pre-die incubation that in the original protocol is 48 hours. We went lower and also higher. And we noticed that we have with an increasing time of incubation, we reduce the background noise. So this is an example of some of the conditions and the results that we obtained. This is the original protocol, and you can see in blue controls and in red the ASD samples. And here we have when we change one parameter at a time. So in this case, we cut in half the number of cells per well. And you can see that there is an increased variability in some of the population. And here we doubled the population of cells. And you see especially the ASD are tighter. In this case, we decreased the percentage of the fetal bovine serum. And you can see that the differences between patients and controls are increased. So we enhance those differences. So then we try to combine some of the parameters. In this case, we moved the incubation from 48 to 72 hours, and we doubled the cells. In this other case, we have again 72 hours with lower FPS and double the cells. And you can see how tight the ASD are and how much the difference has increased with controls. Here we have increased hours and decreased cell numbers. And here finally we have increased hours of incubation, decreased fetal bovine serum, decreased cells. But unfortunately, if we decrease the cells, you see how the error bars are enlarged. So we increase the variability, and we don't gain much in terms of difference with control. So overall, we selected these combination of 72 hours, 1% serum, and 40,000 cells per well. And we tested that over 50 samples from ASD and 50 controls. So the final thing was increasing the number of cells, increasing the incubation, decreasing the serum. And you can see here is the original protocol, and here is the new modified protocol. So for all the wells in the tryptophan plate. And it's striking how much the difference has increased between patients and controls, and also how much is decreased the variability, especially in patients in ASD, and how much the background noise is decreased. So you can see here how the empty well, where we were concerned about non-specific production of NADH that was comparable with the one observed in the presence of tryptophan, has decreased, and so we're now more confident about what we observe in terms of outcome. And so overall, we realized that exacerbating the metabolic conditions, because by increasing the cells for the same amount of energy source, decreasing the serum, and expanding the incubation, we create a more extreme and more competitive metabolic environment. And this leads to reducing the background noise, reducing the variability within the samples, and enhancing the differences between ASD and controls, so increasing the discrimination power of the test. So our conclusions were that basically the ASD cells are less capable of producing NADH in a more challenging metabolic environment as compared to controls. Then we explored other conditions that present with ASD and also other types of neurobehavioral disorders. One of these was the ACTB2 associated syndrome, so-called because it's associated with either pathogenic variants or rearrangements affecting the ACTB2 gene. And you can see here some of the individuals described in a paper from 2017. The common features of these conditions include infantile hypotonia, so low muscle tone at infantile age, developmental and speech delay, several behavioral issues, including autistic tendencies. So this is, like I said, a syndromic form of autism, but also hyperactivity, aggressiveness. Then they have palatal and dental anomalies, as you can see here, some of the prominent teeth. And they also have skeletal issues with some predisposition for bone malformations or fracture. So we applied the same approach. We looked at all the plates, and we realized that, first of all, we were able to see, once again, the reduced tryptophan utilization that was in line with what we observed in cases with both syndromic and non-syndromic ASD. And you can see here the data from the tryptophan plate with the new protocol that showed clear significant differences in all wells containing tryptophan with very low background noise. But when we looked at other wells, we looked at other plates of the platform, we also noticed, and this was performed in 11 patients with SATB2 associated syndrome versus 50 controls. So we noticed some clear differences in the utilization of phosphorylated sugars. You can see here glucose 6-phosphate and 1-phosphate. Which is balanced to some extent with the increased utilization of alternative energy sources like maltose in this case. And this might be related to the growth and developmental delay. These patients are actually smaller than their the children of the same age. This might also be linked to the fact that we noticed an increased response in terms of NADH production when the cells were exposed to several metabolic effectors associated with growth regulation, like iodine and thyroxine, or T4, that are related to the thyroid metabolism. Cyclic AMP, which is associated with is one of the most important secondary messengers, so it mediates cellular signaling, and insulin, that is one of the most important catabolic hormones, so it helps build up our body. So all these profile is compatible with an increase in basic metabolic needs, and that is coupled with the decreased utilization of major energy sources. So you can see that there is a disproportion here in the metabolic profile of these individuals. And also another interesting feature, another interesting feature that we detected was related to the beta estradiol hormone. So as you can see here from the graphic, the asterisks are related to the last 4 wells of the series. There are in fact 6 wells containing beta estradiol on one plate, and the differences are dose dependent because the 6 wells have increasing concentration of estradiol. So it is interesting to notice that with increased concentration, there is an increased response in terms of NADH production. And this may be a possible compensatory mechanism to the lack of estrogen receptor beta activity that might be linked to the skeletal anomalies. In fact, it has been described in a paper from 2020 that estrogens, including therefore beta estradiol, enhance the level of the ACTB2 protein, and ACTB2 is also a target of the estrogen receptor beta. So there is a role for ACTB2 in pluripotency and senescence of bone marrow stromal cells, and it might be related to postmenopausal osteoporosis. So if you imagine removing ACTB2 from that equation, from that pathway, so in the presence of ACTB2 mutations or deletions, we might have a disruption of the estrogen receptor beta mediated regulation of bone growth and stability, and that might be reflected into the skeletal abnormalities described in these patients. And then we explored another condition associated with syndromic ASD, the Phelan-McDermid syndrome, or PMS. This is a condition characterized by very marked clinical variability. We have some features that are shared with ACTB2-associated syndrome, like the neonatal hypotonia, developmental delay, and especially delayed or absent speech. We have, of course, autistic traits. We have other features like seizures, high tolerance to pain, gastrointestinal issues, and kidney problems. But as you can see from the pictures here, we don't have a typical facial pattern. There are some minor dysmorphic traits, but the variability is really the key factor here in this condition. And this clinical variability is matched by genetic variability as well. In fact, the syndrome is caused by various deletions of the 22q13 terminal region. As you can see here, the red bars are showing you the size of the lost portion of the 22q13 region. And you can see they might be very small. The smallest described so far is 0.1 megabases, which means 100,000 nucleotides. And they can go up to 9 megabases, which means 9 million nucleotides. But there are also other types of rearrangements, like ring chromosome 22, translocations, interstitial deletions, as you can see in the top 2 bars here. So deletions that do not involve the telomere. Or there could be pathogenic variants affecting the SHANK3 gene, which is down here. I don't know if you can see it, but it's one of the last genes, so one of the closest genes to the telomere. And it is a very important gene in synaptic stability and formation, and therefore it might be linked to several neurological features in PMS. So in order to address this extreme clinical and genetic variability, we used a double approach. We looked at the epigenetic profile of different groups of patients with PMS. We distinguished the patients by their deletion sizes. So we have patients with deletions larger than 1 megabases, and we call them the large group here in red, and cases with the small deletions, so less than 1 megabase, or pathogenic SHANK3 variants. And they're called the small group, you can see in orange. And it's dramatic how the large group is actually distinguishable from not only the small group, but also 2 different and independent populations of controls. So the small PMS group really did not present with epigenetic features, so with methylation profile features that were allowing us to sort them out from controls. On the other hand, the large deletion group was very different, was extremely different. And we can see that also from this other picture, where you can see the cluster still based on their methylation profile. And the red one, the group with the PMS large deletions, is clustered together and is very much separated from the rest of the samples that include the small deletion group that is interspersed and two populations of controls. So that was one approach, and this approach was highlighting the big difference in terms of episignature, so epigenetic profile. And the threshold was about 1 megabases from the terminus, from the telomere. And we realized that one of the possible explanations for this dramatic difference was the presence of the BRD1 gene that you can see here highlighted from this arrow. And this gene is involved in genome-wide methylation. So perhaps losing one copy of the gene will disrupt the methylation in the rest of the genome and will disrupt the epigenetic profile. But very interestingly, the metabolic profile that we also tested using the biolog arrays was matching these differences. And in fact, when we looked on the same cohort of 11 individuals with large deletions versus 9 individuals with small deletions or SHANK3 mutations, we noticed that the small group only had one well of all the 8 biolog PMM plates plus the tryptophan plate. Only one was significantly different from controls, while the large group had a series of wells that include, among the others, wells involved in the energy production. So there was a reduced utilization of main carbon-based energy sources. You can see here the phosphorylated sugars, glucose, and mannose. Also reduced utilization of some key Krebs cycle intermediates like pyruvic acid and alpha-ketobutyric acid. Reduced utilization of large amino acids, including tryptophan, as described not only for the tryptophan wells, but also for other amino acids of the same group. And there was also reduced response to growth factors like the human growth hormone and fibroblast growth factor 1, and reduced response to cytokines. So all these metabolic features were in fact associated with some clinical features reported in some patients with PMS. For example, the energy problems might be related to neurodevelopmental delay, where the problems with response to the cytokines like interleukin-1 beta may be related to the predisposition to recurrent infections and chronic inflammation in these patients. So overall, to wrap this up, we realized that by using the metabolic approach based on the PMM assays, we were able to address some of the challenges that I mentioned at the beginning. In particular, we were able to provide a functional investigation of pathogenic mechanisms, for example, the link between tryptophan and ASD, or the possible pathogenic mechanisms in cases with PMS carrying large deletions of chromosome 22q13, or the link between estrogens and the bone problems in SATB2 associated syndrome. We also had, we were able to collect data that were very important for potential development of biomarker screening tests. For example, the new protocol for the PMM tryptophan plate with improved discrimination power might be very useful, not only to provide a lab test for ASD, but also eventually to screen individuals at high risk before the presentation of the symptoms. So if we think about the average diagnostic age, which is 3 years old, the metabolic profile of these individuals must be detectable, and we did have some early cases at a very early age. So it might help at least to identify individuals, for example, in families where we saw there is a high recurrence risk that might actually have a higher risk for developing ASD. And finally, this approach allowed us to identify or validate molecular targets for treatment. For example, tryptophan supplements in ASD, or eventually an approach based on dietary supplements that might help the behavioral symptoms in these patients. Estrogens in ACTB2-associated syndrome. Again, it's something that needs to be explored further, but it might be useful for the skeletal issues. Or targeting the BRD1 gene in PMS cases with large deletions because of the possible consequences at the genome-wide methylation level. And with this, I would like to thank you all, and particularly I would like to thank all the people that made this possible. First, the group at the Greenwood Genetics Center where the data were generated, my closest collaborators here, and all the people that I had the privilege to work with, the people at Clemson University, where I am now, and that worked with me on many of these projects, but also my good friend and collaborator, Yuri Zarate at the University of Arkansas, and Jenny Lee Orsell, and Laila Schenkel and Becky Sadikovic, and also last but not least, the Phelan-McDermid Syndrome Foundation. So thank you all, and I'm ready to take questions.